Alright, welcome back to another edition of Mr. Bentley Does the Review Key. Very excited to have you with us this time. This time I'm doing the exact same thing I've done every time. Please follow along. Remember, skip to the question you actually need to see. You don't need to hear me go through every single question. Most of the time, you'll know everything I'm saying, because I try to make these review keys for people who don't know anything at all about the question they're on. So, hopefully, you don't need to watch the whole thing. Alright, that said, I'm now going to stop talking like a crazy person and get to the review key. Thank you. Alright, so the first question is, your phenotype is determined by which genes are blank. And you look up at the answer choices at the top. Phenotype is a physical trait. So let me just type that in there. Remember, that is what you actually express. Generally, it's going to be a protein being produced or not produced. So, when we produce a gene, or a protein, that is called expression. So, expressed. Alright, now we'll come back to that word in a bit because it turns out it's a little bit trickier than just saying it means proteins being produced. There's a better word that we're going to use. If we say the gene is active, that means it is currently being used to produce proteins. So, active. And so that would also be considered expressed. That's what the rest of that sentence says. On the other hand, repressed genes are those that are currently turned off. So repressed, like a repressed memory, is a memory that you cannot remember because you don't want to. So a repressed gene, it's there, but it's turned off at the time. You're not going to use it. Okay, so gene expression is not just means on. That's what gets confusing. Gene expression has two uses. It means the gene is turned on, or it is simply the process in total. So if I ever say genes being turned on or off, notice I said or, that would be gene expression. If I said a gene is turned on, you would then say it is expressed. So it's a very subtle difference, so re-listen to that if you're having trouble. But gene expression can explain how all cells in your body contain the same number of chromosomes, yet they manage to do totally different things. So remember, chromosomes are the structures of DNA. So every single cell from the tip of your eyeball to the bottom of your foot, yes, very different structures. Same DNA. The exceptions to that, sex cells and blood cells in humans. But they have the same DNA, yet they produce different proteins. That's what's so crazy. Totally different proteins being produced, different phenotypes are the result. And that's it. I mean, we talked about this expression, transcription, translation as the central dogma of modern biology, which is just a nice way of saying really important stuff. This is why we can do so many things. I could go on about this for a while, so I'm just going to stop. This is a key, not me getting to rant. I get excited about it. Number 2, identify the type of mutation shown in the examples below. Determine how would it affect the protein made. Okay, so let me scoot this over so we can see it a little bit better. Oh, too far. And there we go. Alright, so it matches GCCCT, blah, 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 blah. Okay, that was exactly the same. Confused. Oh, there it is. My bad. Right there. I'll underline it. CCC went to CCA. Let's see, that would be GGU. I'll have to go pull up a chart, so let's first say what this mutation is. This is what we call a substitution. So, in a substitution mutation, you take a nitrogen base, and you substitute in a new nitrogen base. So like if a substitute comes into my room, it's replacing me. It's not like I'm there and it's there. You're not missing a teacher. You have simply a substitute. Now, just like with Real substitutes, most of the time the substitute does nothing. Ha ha ha, yeah, poor substitutes. I hope I haven't offended anybody out there. Sometimes the substitute, it doesn't work. Sometimes they just do the normal teacher thing. Hopefully, this is very rare, but sometimes a substitute's better than the original teacher. Again, hopefully that's pretty rare for your class. That's kind of how substitutions are in real life. Sometimes they can't tell, sometimes it's bad, sometimes it's better. That's probably the rarest of them. Okay, so the effect. Now I'm going to have to go get a chart, so I'm going to pause this real quick. Alright then, we're back. Alright, so I've now added a amino acid codon chart to this review, so let's go through it. We have GCC at the beginning. Now what you got to remember is in order to do these, you must first turn it into RNA. So what was GCC is now CGG, then it is AUG, GGU, UCG, AAU. Hopefully I didn't mess any of those up. It's a little tricky doing this in front of people. Yeah, that looks about right. Alright, so here's what we're going to notice. This first CGG, not important. It gets left out. This is the start. AUG. Remember, school starts in August. And if you went up here, you'd find out that AUG is what we call methionine. Alright, so the start will be at AUG. So let's just do that right here. Amino acid start. Alright, GGU. GGU is glycine. Well, the original DNA message was GGG. But GGG, so sorry I did that in my head. GGG is also glycine. So this is actually what we call a silent mutation. Silent mutation of glycine. Because it was originally glycine in the CCC, and it's still glycine. I could do the rest, it doesn't matter, the rest is the same as the original message, and that's significant. That's actually a property of substitutions, because they simply replace without changing the order of the letters afterwards. It ends up being usually fairly clean, one change or no change. So, in this case, it was a silent mutation, which is just fine and dandy. Alright, let's try this again, but this time, let's see, we're changing this beginning. In this case, let's see, CAT, we have an inversion, it looks like. Let me underline the inverted section. Notice, before it said GCC, T, A, C, 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 C. Then it continued, A, G, C, T, T, A. So what happened is, this occasionally happens, a section breaks out and is put back in, but in the wrong direction this time. And that's exactly what has happened here. Alright, so let's say what that is. This is called an inversion. Inversion. And this one's pretty easy because if you look through here, none of the amino acids say TAC, and thus it will never start. So, no start codon. No. That's huge. That's a bad effect. As you may imagine, for a protein that needs to get produced, if there's no place that says start, it doesn't get produced. This is what we call a nonsense. If any time you mess up either the start or the stop of a protein, we say that it is a nonsense mutation. And what that is the start codon is missing. And thus, honestly, you probably have some other start codon somewhere in the long chain of amino acids, and the cell's going to try to make a protein out of that, and it's probably just not going to do anything because you're missing out this whole beginning section until the cell happens to spot an AUG, or sorry, a TAC in the DNA. So the nonsense tells you the start codon is missing, or an early stop codon was made. Okay, let's keep going with that. Alright, this time we have GAC, and let me just copy and paste the original real quick, just so I can have an extra copy and I don't have to just keep scrolling back and forth. Copy. Alright. Alright, so GCC, TAC, CC, blah 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 blah. Alright, this is a fun one. The beginning has been mutated. It looks like maybe a double or a substitution right here. This is the only thing that's been substituted. There has been a substitution. And this one's kind of interesting. This is no effect, or at least none that we can tell from the amino acid sequence. And here's why. Your protein sequence doesn't start till here. And so there was no effect because the mutation was before the start codon. It was in a non-coding region of the DNA. So that's not a bad mutation to have. Alright, so continuing on, we got GCC, TAC. Ooh, this is a bad one. Alright, CCA, CAG, CAG. So let's go see if we can figure out what's happened here. It looks like maybe an insertion. Yes. Alright, so here in the original DNA, this was CCC. And then keep reading. A, G, C, T, T, A. The rest is the same, so the only new section is that A. And that caused a huge shift. Everything has now been shifted over to the right because of it. Thus changing what we call the reading frame. To summarize that, we call this a frame shift. Everything after our insertion has shifted over, just messing all kinds of stuff up. Okay, so let's go through it. This is a probably a missense. Now let me first say that this is a insertion mutation. Mutation is insertion. The effect will be all the protein downstream of the mutation will be messed up. Now, what you have to look for is if was an early stop codon produced. Now, in reality, if this occurred anywhere towards the beginning of the protein sequence, most likely it's going to end up causing an early stop codon to be produced. But in this scenario, you don't actually know yet, so you actually kind of have to check. So let's go through it. Let's see, we start here. Let's do the RNA first. AUG, GUG, GUC, GAA, and we don't need to worry about that last one because we don't know what it is. So it starts at AUG. GUG. Let's see. GUG will be alanine. ALA. GUC, if you remember from back there, was also ALA. Oh, one sec, getting a little bit of lag. ALA. And GAA, let's go back and check that out because I don't know that off the top of my head. GAA is GLU. I think that's glutamine, I can't remember. But when we look at this, it's going to be totally different from the original sequence. Totally different. And that's just because everything after that frame shift is now messed up. So this will be a missense because no early stop codon was produced, but the protein is messed up. The protein has mistakes. Not a nonsense, because no early stop codon was produced. Okay, so a missense will be any mutation, a mutation effect where the protein sequence is changed. A nonsense is a mutation effect. It's like an upgraded missense, where an early stop codon is produced, or the start codon is mutated into not occurring. I guess. I don't know how to say that. Non-existence? That sounds too dramatic. Silent mutation is exactly what it sounds like. It's there, but it's quiet. You can't tell because it doesn't actually end up changing the amino acid sequence. So that'd be like earlier we had, I think it was a glycine that mutated, and yes, the DNA changed, but it still said glycine whenever the ribosome tried to translate it. And remember, to figure that out, what your ribosome would do, you have to go consult our chart of what the ribosome does, the codon chart. So a silent mutation is the DNA changes, but the amino acid sequence remains the same. That's it. And it's due to the redundancy within the codon chart and how our ribosomes produce proteins, or translate. Okay, so in this picture, this is a virus. It's actually a bacteriophage. I happen to know. I don't think you don't need to know that. Don't worry about it. The structure C, though, is pointing at, looks like some squiggly stuff inside, and if you remember these long squiggly chains, are probably going to either be protein or DNA or RNA. In this case, we're looking at the inside of a virus, and thus this must be DNA or RNA, so nucleic acid. DNA or RNA. And notice I don't say and ENA, kind of tricky. Okay, well that's not an easy question. So describe how viruses are able to replicate. So a virus is what's considered an infectious particle. It's like a message gone. It's like a chain letter. It's like a bad message. It's a message that says make more copies of this message. And sometimes they're more complicated than that, and sometimes they're just that simple. But a virus is able to replicate by when it touches a compatible cell. The virus has its DNA inserted into the cell. Let me put DNA or RNA. where it tells the cell to make copies of it and send them out into the environment. Whew. I spelled all that correctly. I'm impressed. Now, there's lots of ways to complicate this. There's some viruses actually put their own DNA inside of the cell's DNA. There's some that don't even bother with DNA. They just inject it with RNA and send that RNA to the ribosomes inside the cell. There's lots and lots of different viruses out there. Seem to be many of them that have evolved alongside the organism which they infect. Like, many viruses are very specific. A human virus doesn't infect any other organisms, or only similar organisms. And and so there's a huge variety, and we're only now starting to really discover them. So I mean, viruses are some cool, cutting-edge kind of stuff. Cutting edge, not age. That was weird. Alright, so what would have a greater impact? A virus infecting a somatic cell or a virus infecting an egg cell? Now, this is a crazy cool question because it happens. Humans have 5 to 8% of our DNA virus in origin, or what we call viral in origin. And what that means is that some kind of stem cell or sex cell of a human being, or some of the ancestors of human beings, was infected by a virus and then passed that virus DNA into all of its children and eventually to us and to you. So we all have this big chunk of our DNA. Remember, our DNA is huge, so 5 to 8% is not a small amount of DNA. 5 to 8% is quite a bit. I just said that. Okay. They have passed it on down to us, and it's just part of us now. We've turned off the sections of viruses. You don't just sit there making viruses every day, infecting the world. No. We've turned off that section, or used parts of it to do certain things. There's lots of interesting ways our cells have adapted to this virus DNA. And again, it's more stuff that we're only now starting to learn about and understand. So, again, cutting edge stuff. Alright, so a virus infecting a somatic cell or a virus infecting an egg cell. Well, an egg cell infected by a provirus, which would be a virus that inserts itself into your DNA, would pass on the DNA to the entirety of its offspring. And that's big. Thus changing the DNA for that species. If it continued to go into if it continued to be able to reproduce. Now, I imagine most of the time this happens, the baby just aborts. It stops developing. Because the mother wouldn't want to spend all this energy developing a baby who won't work or is in fact harmful. So generally our bodies try to prevent this kind of stuff from happening, but if the baby that was being produced managed to mutate the virus DNA in such a way that it no longer worked, well, then it would just have this huge chunk of DNA from the virus that did nothing. And so it would just keep developing, and maybe it could use parts of that virus DNA. And so all sorts of interesting ways of evolution being able to occur can occur through these viruses. It's a very, very cool question. Okay. Population genetics. Population genetics is the study of allele frequencies within, it should be one word, a population. The allele frequency is simply a measurement of how common certain traits are in a population. So that's what frequency means, is how common something is. Many things can change the genes of a population. That's true. Now, remember, the original change has to come from a mutation, but then the way those mutations are distributed throughout the population will have a bunch of different names. So random changes, just caused by random events, are called genetic drift. So sometimes certain mutations become favored for no particular reason. There's a fire in a forest, and, well, most trees are flammable, so it's not really going to have to do with your genetics. It just happens to be if you're in the wrong place at the wrong time. You could wipe out a whole branch of, like grandchildren of a tree long ago. They could all be wiped out if a whole section of a forest is burned down. And it had nothing to do with how successful those trees were. They just happened to be in the wrong place when lightning struck and set it on fire. So now, of course, there are some things that aren't random change. We'll get to those in a sec. All right. So next should be a separate sentence. While changes, there we are. While changes due to migration of individuals between different populations is called gene flow. So we all have heard of migration before. Well, things migrate. And when they do that, they take their genes, maybe from very far away, and introduce them to new populations. Imagine, fish take a different route, end up getting lost, and end up breeding with a whole new group of fish. And, that can spread genes around that way. Again, this isn't a natural selection process because it wasn't that they got to spread their genes because they were better. They got to spread their genes simply because they moved. Now, in the end, that may cause natural selection, but that's not necessarily natural selection by itself. Okay, so that's this next sentence. It's changes that are caused by the fitness. So let me define that for you. The ability to reproduce, or the ability to produce successful offspring. Of the trait are caused by natural selection. Okay, so now that has a very specific definition. It is whenever you have changes to a population that is caused by natural selection. So if I go back to my fire example, if you have trees that are especially flammable and they burn more than the non-especially flammable trees, well, that's natural selection. Nature is selecting against trees that are especially flammable. That's why in places with lots of lightning, you don't find trees that just easily burst into flames. And yes, there are some trees that are more flammable than others. So that's natural selection. It's survival of the fittest kind of thing. Blank populations tend to be more greatly affected by genetic drift. That will be smaller populations. Now, you did a whole lab on this. That was that Fruit Loop lab. But basically, the smaller a population gets, the more likely just random genes will be left out of that grouping. I told all my students that if you imagine like one side of the classroom caved in, well, if everybody in there had to survive and had to make descendants, well, they'd probably look slightly different than if the whole class had survived. They'll probably be more similar to each other with the smaller population simply because there's less variety, or what we call a smaller gene pool in a smaller population. So that's the idea. Basically, there is a smaller gene pool in smaller populations, and thus genetic drift can really strongly affect them. Okay, so while blank populations tend to have more genetic diversity, larger, of course. We call that genetic diversity gene pools, is fairly commonly used. Alright, identify if the genetic change is due to gene flow, genetic drift, natural selection. Forgot to do one, artificial selection. When we get there, we'll get there. Or a combination of the two. When the environment determines how successful a trait is in a gene pool. That is natural selection. Straight up definition. The introduction of new genes as new organisms are introduced to a population. That is gene flow. Now notice we didn't say anything about successful genes. We just said new genes. So no natural selection yet. Okay, one sec. My computer's being silly. Alright, random chance events can shift gene pools. That is genetic drift. An easy way to remember the difference between drift and flow is flowing is smooth. You know, going from one place to another, not stopping, you're flowing, which is cool. Random events that shift gene pools, so just random stuff is drifting. Like if you see somebody driving crazily on the road, like going back and forth between lanes, kind of like veering off to one direction, suddenly correcting themselves, we call that drifting. Because it's just random movements, a drift. There you are. So anteaters in a certain population mostly have medium-sized tongues. However, a new anteater moves into the area. Bam, gene flow, right there. That has a longer tongue that helps it find food faster. That is natural selection. Yeah. And there you go. So, natural selection and gene flow. Natural selection and gene flow. Okay, people are going to make fun of me for that one. So, I finished explaining that. Yeah, so you had the anteater moving into a new area, so that was new genes from a first population moving into a second population. Totally gene flow. Then, because it carried beneficial genes, its descendants did better. Natural selection. So both of those. Most ladybugs have black dots on their wings. However, on a trip to Europe, a tourist discovers a population of ladybugs with no black dots. He put several in a jar, brought them home, and released them into his garden. Okay, so, this is a guy who it was a guy, right? Oh, no, it was it could be guy or girl. Oh, no, there he goes. Pronoun, he. So, this guy takes them, brings them back to the United States. So we have some gene flow happening here. It will cause a selection, or it will cause a change to genes, and it's by a human. So that is what we call artificial selection. So, if he continued to do this, he could cause more ladybugs in his area to be born with no black spots. So that is our definition of human selection, favoring a certain trait because of humans. And he caused gene flow to occur if they managed to breed with the current population, which doesn't actually say they did, but we're going to assume they did. Alright, a population of squirrels lives in an area with dark-colored plants. Most of the squirrels have light-colored fur. So one day, a mutation in an individual gets darker fur. Well, he's going to survive better because he's better camouflaged. So that was nature, so the environment of this squirrel, choosing that his DNA is better. And that is thus our definition of natural selection. A lot of people miss that question. I've seen on the quizzes. It was upsetting. And we're frozen again. Give it a sec. There we go. Alright. Let's pull this down. Okay. I think we'll just keep going from there. Brown and white rabbits are moved into... Okay, that was weird. Brown and white rabbits are moved into the U.S., from the U.S. onto a small island with no rabbits. The ship contains crates with mostly white rabbits, and now the population of rabbits looks very different. Okay, so let's see. Was it on purpose? The ships contain crates with mostly... It doesn't actually matter if it was on purpose. Humans have caused this. And so this is going to be a... yeah, this is going to be, first of all, artificial selection. You could probably argue gene flow. Generally, gene flow, you have to have a original population to be affecting. Really, this is genetic drift. So I'm going to say this is genetic drift, where the original population of rabbits is having genes affected by what we call the founder's effect. So these rabbits are moving onto the island, they're establishing their own colony there, but because it was only white rabbits, the population on the island is got mostly white rabbit genes now. So, because of the founder's effect. There we go. Alright, moving this down. Alright, a fire destroyed most of a Texas forest. Wonder what that's like. Just kidding, it happens all the time. Due to the random path of the fire, random path, bam, genetic drift. As soon as you see that, bam, that's what you should think, genetic drift. Nothing about natural selection, nothing in here has to do with its better fitness, the ability to produce more offspring, that's not in here. Nothing about humans, no movement of organisms, genetic drift, that's it. Alright, so let's add a few indentions, move this down here, and see what we're doing. Okay. So we are defining the following words. Okay, I can do that. I don't know what the word bank's for. I guess that was for the last question. I'm just going to take that out. Evolution. Change in gene frequency over time. Easy enough. You know, any kind of changes in gene frequency over time is causing evolution. Sounds good to me. Gene pool, the total variety of genes in a population. The broader you define your population, the bigger your gene pool. The smaller your population, generally the smaller your gene pool. Yeah. Genetic drift is random changes. Oh, slowing down. One sec. There we go. Random changes to gene frequency. Has nothing to do with fitness. It has nothing to do with fitness. Usually caused by natural disasters. Often caused by natural disasters, but many other things. But if you remember that one, you'll be pretty good. I think I'm using singular, so I have to say a singular possessive. Oh, be quiet. Nobody cares about grammar. Said never Mr. Bentley, because he is on good terms with most of your English teachers. Gene flow is whenever organisms move from one population to another and thus change the genetics of their destination population by introducing new genes. So when organisms move in or out of a population, changing gene frequencies. Population genetics, we literally just defined that a few minutes ago, but it is the study of gene frequency changes in populations. Thus, it's the study of evolution. I don't know. I'm sure somebody would get mad at me for that definition. Just go back up to the upper one. You can do it there. Get it from there. Virus is a infectious particle. It is considered non-living. Non-living because it does not do anything besides... I should say, it does very little. People get mad at me for that. Because it does not use energy. or have a cell non-cellular. It does not adapt to its environment. Pretty much the most it can ever do is inject the proper cell with its DNA or RNA. But here's the big one. Cannot reproduce without a host cell. That's the huge one. So that is all the reasons why those are a lot of the reasons why it's considered non-living. But there's more you need to know about it. Nucleic, simplest variety are DNA or RNA surrounded by a protein coat called a capsid. And that's about as much as you need to know. There's so many more types, but honestly, we don't know all of them. They're so hard to observe, they're so hard to grow, they're so hard to find that it's simply constantly developing, and we're constantly learning new things about them. They're just like this whole other branch of life-like things. And really, it just has to do with the fact that we happen to cut off living at those above conditions. So, there you go. Alright, mutation. So, mutation is change in DNA. Bam, easy. In DNA. Or RNA. How about we just go with genetic information? Okay. Gene expression. Genes can be turned on or off to produce different traits. Okay. 8. There we are. Am I done? Sweet. Alright guys, I hope that helped. Please ask me questions, send me emails with whatever you need. My name is Nolan Bentley. I'm in room 1212 across from the Academy Principal's offices. I have tutorials Monday, but your test is on Tuesday, so you better get in if you need to. Anything else? At the bottom in the summary, I will include links on to this original review, and also a link to the completed key. I'll probably go back through and kind of reformat it a little bit so it's prettier. But there you go. My email is nolan.bentley@cfisd.net. So nolan.bentley@cfisd.net. Alright guys, have a great day. Thank you.